Imagine trying to build a shelter in the middle of a chaotic storm. You need walls strong enough to keep the elements out, but you also need doors to bring in food and take out the trash. Every living cell faces this exact same problem. To survive, a cell acts a bit like an exclusive nightclub. The boundary remains flexible, while bouncers at the doors check who gets in and who stays out. Biologists call this property selective permeability. Look at this cross-section of an actual cell membrane. The boundary consists of two layers of fats, known as a lipid bilayer. The heads of these lipid molecules point outward toward the water, while their tails hide on the inside. This structure moves constantly. The lipids shift sideways and flow like a liquid sea. That's why the first half of its scientific name is the fluid model. Floating inside that moving sea are various proteins. Because these proteins are scattered throughout, like tiles in a mosaic, we call the whole structure the fluid mosaic model. These proteins act as the cell's bouncers, creating channels that allow only specific small molecules to pass through. By combining a flexible lipid sea with strict protein gatekeepers, the cell is able to maintain a highly controlled, stable internal environment, no matter what is happening on the outside. One of the most important molecules that needs to get past that boundary is water. The specific process of water molecules slipping through a selectively permeable membrane is called osmosis. To see how osmosis works, we need to compare two different environments in this diagram. On the left side, we have pure water, 100% blue water particles. On the right, we have a concentrated salt solution, mixing water with large white salt crystals. Here is where students often get trapped. When you hear high concentration salt solution, it's easy to assume everything is highly concentrated. But those large salt crystals take up physical space. That means a high salt concentration actually results in a very low water concentration. This illustrates the governing rule of osmosis. Water moves from a region where there is more water to a region where there is less water. If you get distracted by the salt or sugar in a problem, you will likely predict the wrong outcome. Always track the water concentration first, and the rest becomes simple physics. Let's test that rule. Here we are dropping cell X into a beaker of pure water. Notice that the environment outside is packed tightly with water particles, while the inside of the cell has a lower density of water. When we compare the two, the disparity is obvious. We have a higher water concentration on the outside and a lower water concentration on the inside. Following the rules of physics, water flows from high to low. It rushes through the membrane and into the cell, pushing against the flexible boundaries and causing the whole cell to swell up. Because of the pressure of osmosis, anytime you place a cell into an environment that is purer than its own interior, it will take on water and expand. Now let's try the opposite scenario. We're going to drop a new cell, cell Y, into a concentrated salt solution. Before we drop it in, use the rule of water concentration to predict the outcome. Will cell Y swell or shrink? Pause the video if you need a moment. Let's look at the environmental data. Because there is so much salt taking up space in the beaker, the water concentration outside the cell is much lower than the water concentration inside. Once again, water moves from high to low. It travels outward, escaping the cell to reach the lower concentration in the beaker, causing the cell membrane to shrivel and collapse. The cell doesn't choose to shrink. The external salt simply takes up space, forcing the internal water out to balance the scales. We can see this exact process outside of the microscopic world. Take a handful of plump, water-soaked mung bean seeds and drop them into a concentrated salty brine. Watch this time-lapse over a 12-hour period. Just like cell Y, the high salt concentration outside means there's less water outside. The water particles exit the beans, leaving them wrinkled and dehydrated. Let's build a final summary. First, the boundary itself. A lipid bilayer combined with embedded proteins gives us the highly flexible, selectively permeable fluid mosaic model. Second, the rules of osmosis. Place a cell in pure water, and water rushes in, making it swell. Place that same cell in salt water, and water rushes out, forcing it to shrink. By understanding the fluid mosaic boundary and tracking water concentration, you can accurately predict how materials transport in and out of any living cell.